Okay, good evening. We will wait for a couple of minutes before we start. I'll just download the slides and get my iPad connected. Just give me a moment. I hope my screen is visible. Okay. Okay, today's session 11 of the 12-week course, Enzyme Sciences and Technology. And today's session is about enzyme applications. So we will talk about the applications of enzymes, and this is part one. In the next week, we will be discussing about part two. And just to before I begin, a brief introduction. This course is about enzyme science and technology, which is taken by Professor Vishal Trivedi of IIT Guwahati. I'm Sanket, research fellow from the Kusuma School of Biological Sciences, IIT Delhi. And let's get into the topic. So, so the applications of enzyme, if we look at it, give me a moment. Okay. So the applications, if we talk about the applications of enzyme, so I hope this is visible. Is it visible? So talking about the applications of enzymes can be used for drug delivery. They can be used for agriculture. And we will see how they are used. For pisciculture, that is for culturing fishes. They can be used for poultry. They can be used for vaccine, transgenic animals, medicine, and also to form genetically modified organisms. Right? So these are the uses of enzyme. Now talking about, just give me a moment. Now, talking about the enzymes, the broad application of enzymes, we can say they can be used for, let's say, for food biotech industry, for recombinant DNA technology, for medicine, obviously, and for disease biology. Fine. So, uh, so in case of food industry where enzymes can be used, it can be used for, let's say, poultry, right? For fishes, culturing fishes and other things like wineries, breweries, stuff like that. There we use enzymes very routinely. So we'll look at it. So before we understand how enzymes are used, we need to know what is the food production chain, right? And food production chain, uh, in terms of the commercial food that how we get it, is initially we have the production of food that is done you in farmlands and things like that, normal production. Then we have processing of this food. And then we go for the distribution of this food. Now, this distribution can be of two types. They can go for retail distribution, where it goes for the end consumer is, users usually, or it can go for restaurant based, distribution. There are other distribution methods, like they could be wholesale and all that kind of things. So the main thing is retail and restaurants are the main consumers. From retail, we mean the home users, and the restaurant businesses, they have mainly, they are the end users of this food chain. Now, talking about the processing, so talking about food processing, so it combines raw food ingredients to produce a marketable food product. Fine. Um, and it is then sold to a consumer. Its final destination is to a consumer. Now, if we look at food processing is not what is the aim of this, but what we, if we look at the enzymes that are used in case of foods. So for, let's say, for dairy production, we have certain enzymes. For dairy products, we have enzymes like rennet, then we have lactase, then we have protease, which is usually in most of the enzymes, I mean, most of the food products. Then we have catalase. Similarly, for other kind of products like brewing, that is for making beer and wine, we have beta glucanase. And we'll talk about all these enzymes: alpha amylase, protease, and amyloglucosidase, right? Similarly, if we see baking, again, a kind of food industry, very prominent one, baking, we have maltogenic amylase, we have glucose oxidase, and uh, pentosanase, stuff like that. So these are in baking. So in wine and fruit juice, we have pectinase and beta glucanase. Then for meat, we have protease, and papain. So these are all the enzymes, right? So talking about the first enzyme, so rennet is extracted from the fourth stomach of young cows, right? It contains enzyme that is required for milk to become cheese. So that happens due to the enzyme rennet, right? So it also separates liquid whey from solid curd, and different animal rennet, usually in cattle only they are found, is used for production of different cheese, right? And the most common vegetable rennet is called thistle. Must have heard about, come across this term. So then we have lactase. It can be artificially from yeast, right? It is required for digestion of whole milk. So of course, lactase is an enzyme which is used to convert, um, lactose. So it will digest lactose. So for creation of lactose-free milk, lactase is used. So because a lot of people are lactose intolerant, so for that, lactase enzyme is used to remove lactose. So similarly, not only lactose-free milk, but also ice creams and other dairy products are also made. Let's say condensed milk also. They do not have lactose in them because of the enzyme lactase. Now, catalase is another enzyme again used in dairy industry. So catalase is produced from bovine liver or microbial sources. So it breaks down hydrogen peroxide to water and oxygen. So that's the function of catalase. And so what happens, along with glucose oxidase and catalase, these two are used to treat the um, wrappers that will prevent food's oxidation. It will prevent the food to get damaged. So for that, we use catalase along with glucose oxidase, right? And even in any cases, so this is for this, in any case, to remove hydrogen peroxide from the system, catalase is usually used. In case of, let's say, cold sterilization, uh, catalase is used, right? So now talking about another enzyme that is protease, very common enzyme. Let's talk about it. So protease is it's widely distributed in the biological world, and it hydrolyzes specific peptide bonds. Right? It hydrolyzes specific peptide bonds, and macropeptides in production of cheese. It's usually, if you're talking about dairy industry in specific, it gives a bitter flavor to the cheese. That is due to the action of protease, and these are peptidases or proteinases. These are some other terms for proteases. These are a complex group of enzymes that can hydrolyze the peptide bond. Now, talking about protease, they can be of, again, three types. Based on the site of action, the functional group, present on active site, or the pH at which they function. Right? So based on the site of action, they can be exopeptidase or endopeptidase. Based on the functional group, they can be, let's say, like serine protease, cysteine protease, something like that. pH for that, there could be alkaline protease, acid protease, or neutral protease based on the pH. Now, proteases are potential targets for developing therapeutic agents against fatal diseases such as cancer or AIDS because of its involvement in the life cycle of the causative organisms. Now, if we talk about sources of protease, there could be different sources like bacteria or fungi, and then there are multiple types of proteases which it uses. Proteases are mainly used, which are commercially used in case of the detergent industry or the leather industry, food industry, and things like that, and also pharmaceutical industry. Right? And these proteases they can be produced through two ways. One is the submerged fermentation. You can read about these in the slides that are there for you, or there could be solid state fermentation, right? Uh, so in solid state fermentation, what we use the carbon source like molasses or wheat brands, soybean brands, these kind of things, which are solid. Whereas in submerged fermentation that happens in a batch reactor setup. You can read about the different factors. For example, if we talk about solid state fermentation, the factors that affect solid state fermentation is, let's say, the moisture of the substrate. Then there is the water activity, incubation temperature, and let's say heat and mass transfer effect. Fine, things like that. Now let's go to the next industry, the brewing industry. So for that, we have already said that there are proteases again used. So these proteases, what they will do, they will break large proteins and enhance the retention of beer, and it will also reduce the haziness of the beer. Okay. In malting process also, proteases are used in the process of malting, which is common in case of beer production. Now, then there is beta glucanase. Beta glucanase is it breaks down carbohydrate bonds within beta glucan. Right? And it aids in filtration after mashing. Okay. Alpha amylase. Alpha amylase is used to it converts starch to dextrin. Also known as limit dextrin in some cases because it's the last, because process, and it is used to in production of corn syrup. It is used, right? It solubilizes carbohydrates found in barley and other cereals, and it decreases the time required for mashing. If we look at the applications of alpha amylase in specific, um, right? Starch, detergent, even in the paper industry, textile industry, and baking industries. These are certain industries where alpha amylase is used. And starch processing industries use alpha amylase as a substitute for acid hydrolysis in the production of starch hydrolysates. Enzymes used for removal of starch in beer, fruit juice, or from clothes and porcelain, and also used as an anti-staling agent to prevent retrogression of starch in bakery products. Now, in case of the baking industry, let's see what are the some important enzymes. So maltogenic amylase, then we have the glucose oxidase, and then we have the pentosanase. And these are the enzymes that are important. So maltogenic amylase is a flour supplement, right? It has an anti-staling effect, prevents your food to get stale. It modifies starch while most of the starch to gelatinize, and resulting starch granules become more flexible during storage. So that is the function of maltogenic amylase. Now talking about glucose oxidase. So glucose oxidase oxidizes glucose and produces gluconic acid and hydrogen peroxide. So this is the function. Glucose is oxidized to gluconic acid and hydrogen peroxide. Now, hydrogen peroxide is a strong oxidizing agent that strengthens the disulfide and the non-disulfide crosslinks in the gluten. Good working condition help proper functioning of the bakery system. Now, talking about pentosanase, its exact mechanism is not known. It improves the dough's machinability, yielding, and it makes the dough more flexible. If you're talking about bakery, it makes it easier to handle. The dough is more stable and gives better oven spring during baking. Now, talking about the wine industry. If we talk about it, there are two main enzymes. One is pectinase. So pectinase prevents pectin from forming haze and hence to get a clear solution. So this is for improving the clarity of the solution, because if you take out fruit juices in general, if they are not processed, they will be turbid. They will have other components of fruits which will not make it clear. But when you get juices which are commercial, they will be very clear. So that is done using the enzyme pectinase. It is also used for extraction of color and juices. It breaks down pectin and releases methanol. So the function is it breaks pectin, releases methanol, and methanol is very toxic in high amounts. So only a little bit of this enzyme needs to be used because it's hazardous. So seven different enzymatic activities that contribute to the breakdown of pectin from a variety of plants are known. It's widely used in juice and wine processing. The source of pectinase is the fungus Aspergillus species. So that is the main source of pectinase. Now talking about beta glucanase, another enzyme. So beta glucanase accelerates all the mechanism links linked to maturation of leaves. It reduces the maturation duration and improves clarification and filtration and improves the act of the fining agent. Now coming to the another industry, that is the meat industry. In meat industry, two main enzymes again, protease is used, and we have read a lot about protease. It cleaves the bond that between amino acids, we know that, that is the peptide bond. So it cleaves the peptide bond. Just give me a moment. Right? So it loosens the muscles and tenderizes them. So when we talk about tender meat, that comes from the action of protease. Another enzyme used is papain. It is derived from papaya, and that's why the name is papain. It is used in 95% of meat tenderizers, which are available in grocery stores. They are actually papain. This particular enzyme. It is extracted from the latex of papaya fruits. And enzymes are purified and sold in powdered or liquid form. Now, so to conclude, basically from here, these are all the enzymes that are used in the food industry. So enzyme-based processes are advantageous as they can carry out the activity at a moderate reaction condition. So otherwise, this reaction would be very harsh, not very suitable for biological systems. So just to make that reaction condition more moderate, enzymes aid in that function. And in food processing, the market of enzyme is increasing. So these are the two main conclusions from this. Now coming to the enzymes in medical field. So in medicine, enzyme is mostly used as a diagnostic marker, and we will see how it works about certain enzymes, right? Um, so we measured the serum levels of certain enzymes, and it has some clinical significance. It can be organ specific, also giving you the specific problem with a particular organ. You can actually check that. Um, it can be an indicator for tissue damage or cellular damage. So what are the commonly acid enzymes? And we will talk about these. So commonly acid enzymes are like the aminotransferase, of which there is ALT, AST both, which was earlier known as SGPT and SUOT. Then there is lactate dehydrogenase, there is creatine kinase, and gamma glutamine transpeptidase. So these are certain enzymes which are assayed to identify the situation, the physiological condition of a person. Now, talking about alkaline phosphatase, the very first enzyme, right? Alkaline phosphatase, also known as ALP, right? So it's a group of isoform which hydrolyzes many form of phosphate esters. The major source of this enzyme is liver, bone, or kidney, at least in humans. Now, in humans, if we look at this enzyme, elevated level of this enzyme, it actually indicates bone and hepatobiliary diseases. Also because most enzymes are linked to liver somehow or the other. That's why many enzymatic assays actually point towards liver function. But anyway, that's not the point. Apart from liver, also other organs can also be assayed. The condition of other organs can also be determined. It has diagnostic value in hepatic and bone disease in dogs and cats. So that's all about alkaline phosphatase. Then there is creatine kinase. So creatine kinase is the it's the most organ specific serum enzyme in clinical use. Okay. It catalyzes the reversible phosphorylation of creatine by ATP to form creatine phosphate. It's a major storage form of high energy phosphate. In muscles, right? And creatine kinase are found in many parts of the body like heart, brain, skeletal muscle, and smooth muscle. In humans, especially in humans, creatine kinase levels are associated with myocardial infarction, that is heart attack, and muscle diseases. Increase in creatine kinase in cerebrospinal fluid of dogs, cats, cattle, and horses is associated with multiple disorders. The normal value of creatine kinase is around 10 to 15 units per liter of your blood, right? Now coming to another very frequently assayed enzyme, that is alanine aminotransferase. Also known as ALT. This was previously known as SGPT, that is serum glutamate pyruvate transaminase. It catalyzes the reversible transamination of L-alanine and 2-oxaloglutarate, sorry, oxoglutarate. So it catalyzes the reversible transamination. So this is a transamination reaction into pyruvate and glutamate, right? So that is what it does in the cytoplasm. So ALT can be found in liver, skeletal muscle, and heart. It is a well-established indicator of liver-specific damage. Therefore, high levels of ALT or SGPT will actually indicate towards liver damage. And it's not only ALT, also AST, which we will study, aspartate aminotransferase, also indicates towards liver damage. So it's an indicator of hepatopathy and toxicological studies. Its normal values ranges from 0 to 41 units per liter. Now coming to another very liver-based enzyme is aspartate aminotransferase, or AST. It was earlier known as SGOT, serum glutamate oxaloacetic transaminase. It will actually transaminate L-aspartate, very similar to the previous reaction, plus 2-oxoglutarate to oxaloacetate and glutamate, right? It is a good marker of soft tissue. Okay. So AST is actually seen in multiple tissue types. So in kidney, liver, skeletal muscle, heart, and also erythrocyte. Everywhere you would see AST levels. It's found, right? So it's a good marker of soft tissue condition to understand how they are, but it precludes organ specificity, right? Because you do not know which particular organ is affected. Its normal levels are very similar to the previous one, um, 0 to 45 units per liter. Then we have something called sorbitol dehydrogenase. Sorbitol dehydrogenase catalyzes the oxidation of D-sorbitol. It oxidizes to D-fructose. So these enzymes are, again, as I told you, as we have already discussed in the very beginning of this course, that enzymes also have specificity towards chirality. So they only act upon the D form of carbohydrates, so D-sorbitol, and it also produces the cofactor NAD. So it is found in appreciable amounts in liver. So increase in plasma SDH levels indicates liver damage, right? It's very specific towards liver. But in case of humans, we get this test for liver, but for veterinarians, usually ALT is much more standardized, so SDH is not performed. Then there is lactate dehydrogenase, another dehydrogenase. Lactate dehydrogenase causes reversible oxidation of pyruvate. The reversible oxidation of pyruvate to lactate, which is in the L conformation, right? In the LEVO conformation. Now, it again forms the cofactor NAD plus, which is very similar to the oxidation reaction of sorbitol dehydrogenase, similar kind of function. The substrate is different. Now, the equilibrium, it favors the production of lactate. However, due to do assay, we check the direction of pyruvate, because pyruvate is an inhibitor of lactate dehydrogenase, right? And this is important. Otherwise, if the substrate forms a product that enhances the enzymatic activity, the enzyme essentially does not stop its function. So it's very difficult to assay its activity. So although this way is more physiologically relevant, but since it's a reversible reaction, um, the reverse way is actually assayed, right? So LDH is an isoenzyme. It's found in heart, liver, erythrocytes, skeletal muscles, platelets, and lymph nodes. In humans, it is associated with myocardial infarction, hemolysis, and liver disease. LDH profile was one of the first tests which was done in clinical veterinary medicine. The normal levels of LDH is around 60 to 120, sorry, 200, 60 to 200 units per liter. Similarly, we have choline esterase. This is specific to acetylcholine. That's the substrate. Its normal value ranges. It's very small because anyway, acetylcholine is also present in a very small amount, 5.17 units per ml. Right? Then we have the lipase. Lipase is usually from pancreas, they are secreted. So we have serum pancreatic lipases that hydrolyzes the triglycerides. So its lipases actually act upon lipids. So they can be found in pancreas and hepatobiliary tract. Its normal unit is less than 150 units per liter. Similarly, we have assays for amylase. So we have already seen these enzymes working in different industries for their specific purpose. But when we are talking about medicine, we actually want to see if these enzymes are present, and it would actually mark or hint towards some sort of a disease. So it's a calcium dependent metalloenzyme. It hydrolyzes complex carbohydrates. So the product which is formed due to the action of amylase on amylose is actually maltose and limit-dextrin. It's found in the salivary glands, pancreas, and ovaries. Its normal range is from 30 to 110 units per liter, right? Similarly, we have glutamyl transferase. The last enzyme we will be discussing about. It's a carboxypeptidase which cleaves C-terminal glutamyl group. It is associated with glutathione metabolism, and its major source is found in liver and kidney. Again, it can indicate towards hepatobiliary diseases and alcoholism. Its normal values ranges from 0 to 30 units per liter, right? So that's about it. So now we will be discussing certain questions. The rennet in the cheese making process separates milk into cream and skim milk, separates solid curd and liquid whey, enhances the flavor of milk, or reduces the fat content. What would be the right answer? So we've already talked about it. The rennet in cheese making process actually separates solid curd and liquid whey. The most common vegetable rennet used in cheese making is spinach, kale, thistle, or broccoli. So the answer to this would be thistle. Again, we've discussed this. The primary role of lactase in the production of lactose-free milk is to enhance the flavor of milk, increasing the protein content of milk, adding sweetness to the milk, or to break lactose into simpler form. So definitely lactase will work on lactose. The catalase in the baking industry benefits as it increases dough elasticity, prevents yeast fermentation, enhances gluten formation, or neutralizes hydrogen peroxide in dough. Actually, it acts on hydrogen peroxide. So sorry. The technique used to identify various proteases in crude cultural extracts through electrophoresis is polymerase chain reaction, gelatin zymography, gel electrophoresis, or chromatography. It is actually gelatin zymography. The enzyme converts starch to dextrin in producing corn syrup. Is it alpha glucosidase, cyclodextrin glycoside transferase, alpha amylase, or maltogenic alpha amylase? So it's alpha amylase. The industrial application of maltogenic amylase is anti-staling effect in bakery products, juice clarification, oxidizing glucose, or breakdown of pectin in fruit juices. So the answer would be anti-staling effect in bakery products. The enzyme source that produces pectinase, which are used for the removal of inner wall of lotus seeds, garlic, almond, and peanut, is Fusarium oxysporum, Saccharomyces cerevisiae, Pseudomonas solanacerium, or Aspergillus species. So pectinases are usually from Aspergillus species. The action of protease on protein is accelerating biological mechanisms, breaking down starch molecule, loosening muscle fibers and tenderizing the meat, and improving filtration and clarification. So in meat industry, it's used for loosening the muscle fibers and tenderizing the meat. The enzyme that is predominantly found in papaya and used in most grocery meat tenderizers is papain, protease, beta glucanase, or amylase. So it's actually papain because that's how the name has come from papaya. The reason for measuring serum levels of various enzymes in clinical setting is to monitor dietary intake, to identify cellular damage and its consequences, to regulate metabolic function, or to measure the hormone levels. So it's usually to identify any kind of cellular or tissue or organ damage and its consequences. The condition in which the creatine kinase is associated with humans is respiratory disorders, gastrointestinal disorders, myocardial infarction and muscle diseases, or neurological disorders. So creatine kinase is so in humans for myocardial infarction and muscle diseases. The primary neurotransmitter acted upon by cholinesterase at the myoneuronal junction is serotonin, acetylcholine, dopamine, or GABA, gamma aminobutyric acid. It's actually acetylcholine. The mechanism that describes the inhibition of peroxidases by binding of phenoxy radicals during phenol oxidation is allosteric inhibition, competitive inhibition, non-competitive inhibition, or suicide inhibition. So we've discussed about these forms of inhibitions in the previous lecture. And this is mentioned in the case study about about peroxidases. So it's a type of suicide inhibition. The peroxidase is utilized in the determination of glucose, uric acid, and cholesterol in biological fluid as they act as a catalyst in the oxidation of these compounds. They inhibit the formation of these compounds. They catalyze reactions of these compounds, or they function as coenzymes in this assay. So they are used, peroxidase is used as a common, uh, uh, enzyme for all these determine the um, levels of these components because they act as catalysts in the oxidation of these compounds. So with this, we are actually done with this week's session as well. So if there are any doubts, you can ask them now. And if there are none, then we will be meeting for the last session next week on 14th of April, same time. We will be discussing about the enzyme applications part 2. So I hope you have a nice assignment. With that, thank you very much, and I'll be ending the meeting.